But anyway, so my name is Kai Wilkins, and I'm with Advanced Cell Diagnostics. I started with the company in 2012, and I will give you a little bit of information about the company in one of the next slides. Maybe some housekeeping in the beginning. All of you are muted, just that we can offer a good audio quality and everybody can understand. If you have questions, please type them in here in the question and answer box in the webinar pane. So when I was approached by Eres Ras from the Münster University in the Zebrafish lab if RNAscope could also be used on zebrafish whole mount, we had no idea. So we have never tried this before, and obviously I'm a scientist. I have a scientist background. I was curious, and we discussed if they would be willing to try this out and develop this, and they spent some time and effort here to make this work, and I think they really created great results, and this is what they will share with you. It reminded me of my early days when I worked for a company called MWG Biotech, and that's about 15 years ago, and I also had a customer who was working on zebrafish, and they shared this little video with me that was shown here on the right hand side, so the early stage development of a zebrafish embryo, the first hours, and I was really amazed when you see how these cells differentiate and develop, and then you finally can see the embryo. You can recognize the form of the embryo. I'm sure on my screen this movie stopped. I don't know if it proceeded in yours. But anyway, it's a very fascinating moment, and I recall that I had this video and I digged it out of my hard disk from 15 years ago, and I wanted to share this with you as a kind of an opener. So I'm really grateful for this great work, pioneering zebrafish whole mount embryo stuff done by Teresa and Azadeh in the lab of Eres Ras. They have published this. We'll present their results in a minute. In the beginning, I would like to give you an overview of the RNAscope technology, and maybe also say 2 words about our company, so that you see where we are coming from. And maybe so we will start with this, and then I will hand over to Teresa and Azadeh so they can share their results and explain what exactly they have done and how they developed this technology to this success. We will then have a question and answer session, and then I will give you an information about our promotion that we decided to do. That will be the last part. So on the next slide now, I give you two or three words about ACD, Advanced Cell Diagnostics. The company is based in the San Francisco Bay Area. Meanwhile, we have more than 800 customers that use the technology, and that includes all the top pharma, but also many leading labs, academic labs, especially in the cancer field. We have many peer-reviewed publications. We own this technology and have patents on it. We want to develop this technology into diagnostics, so we recently got our ISO certification. We have partnerships with Ventana and Leica for automated staining of tissue slides. We have partnerships with Definiance, which is a software company, to analyze the data we create, to analyze the images. We also have a partnership with PerkinElmer. They have the Nuance system that helps you to overcome autofluorescence problems when imaging. And here in the bottom you see that we are working on cancer diagnostics. So next slide, please. This shows you in a nutshell what RNAscope can do. Each of these brown dots you see in this image reflects a single copy of a transcript in a certain tissue, in the tissue context. So here we look at HER2 transcripts in breast cancer. And what we can deliver, we can give you single molecule detection within the morphological context, and in a way this integrates IHC and qPCR. So next slide gives you a bit more about this idea. As you probably know, the majority of markers we look at today, they are derived from genomic discovery, from either sequencing, next-gen sequencing, or from microarray expression profiling. And when you want to validate these candidate markers, you have these two routes available, PCR or IHC. With PCR, you grind and bind the tissue, so you give up the cellular resolution. On fresh tissue, that works fine. However, on formalin-fixed tissue samples, as this is the pathology routine sample, this is cumbersome. It's not easy to do. And IHC, you're depending on antibodies. If you have a good antibody, it's a great technology, but sometimes you cannot find antibodies, and sometimes the antibodies are not specific or not sensitive enough. So RNAscope gets the best of these both worlds. On the one hand side, we can design a probe for any target, for any species, but at the same time, and we also give you this PCR-like sensitivity and specificity, but from the IHC work protocol, we get the workflow. It's a very similar workflow, as you will see, and you obviously get the in situ context, the cellular resolution. Next slide. So what we can address is we can do all the stuff where IHC typically struggles. So we can look at secreted proteins, we can look at non-coding RNA, we can look at genes where there are no antibodies, or genes that are expressed at such low levels that antibodies are not sensitive enough. And on top of that, we also give you the molecular and the morphological context. So to maybe illustrate a couple of examples where this was applied successfully and where this really makes a difference. Here's one example that was shown by Genentech. They were looking into a certain form of cancer, and they're looking at two targets here. One is HEREGULIN, and the other is HER3. And the HER3 gene encodes for the HEREGULIN receptor. So it's a receptor and a ligand model. And there's one form of cancer that is shown on the bottom left image here. You see one of the targets is expressed at the outer part of the tumor, and the other, the ligand, is expressed in the middle only. And on the right-hand side, the lower image, you see that the expression it's shown in blue and red here, and it's all over the place. So here, ligand and receptor meet each other, and this makes a very aggressive form of this kind of cancer. And when you do a qPCR from these two, from you get the tumor, you grind it up, and you a qPCR assay, looks exactly the same. So it's really the tissue context that helps you to understand why does the one differentiate from the other? Why do they behave differently? So the next slide gives you another example of what you can achieve with RNAscope. Here we look at the family of Wnt genes that are expressed in hair follicle. So we have this kind of specificity that we can discriminate between the different variants here. They are very similar, but nevertheless we can distinguish them. You cannot achieve this specificity with antibodies. So here you see that over time and in different parts of the hair follicle, different Wnt genes are active or not active. Next slide. This is an example of a non-coding RNA, PCA3, for example, monitored in prostate cancer. Next slide. So the idea to look at RNA instead of protein, that's not really new. That was first attempted to do in the late 60s. The first publications came out 1969, and it was done radioactively, looking at ribosomal RNA. Over time, people tried to improve the technology, get it more sensitive, get away from radioactivity, all these things. But they never really had a breakthrough. When you boost signal, the technologies typically also boosted background, and it really takes some tricks to boost signal tremendously without giving higher noise, higher background signal. So we have found a trick how to do this, and that is explained in the next series in a short animation. So what we do is we design pairs of oligonucleotides. They are shown here as Z, and they bind next to each other on a target RNA. So these oligos, they do have a target recognition portion that is complementary to the RNA you are interested in, to your target. Then both have a short linker, and then they both have a tail on top. So it's tail 1 and tail 2, and these tails together are used to bind what we call a preamplifier. This is a long single-stranded DNA molecule, and the upper branch here, the upper stem, is highly repetitive, and it can be used to load many amplifiers, again single-stranded DNA, again highly repetitive, and it can be loaded with many labeled oligonucleotides. So in our default assays for formalin-fixed tissues, this label is a conjugated enzyme like horseradish peroxidase or alkaline phosphatase, and then you can run a chromogenic assay and you get these brown dots or the red dots that I already showed to you. But obviously this label can also be a fluorescent dye that is conjugated to the oligo, and then you get a fluorescent readout. So that's one trick. You see, we bind something that looks like a Christmas tree with many branches and many candles on, and so you get a huge signal. Or the next trick is we not only use a single Christmas tree for any given target, but we load up to 20 of these Christmas trees. And that creates even more signal. We have tested how many Christmas trees do you need to load to see the signal. So when you load 3 of these Christmas trees, the copy shows up as a dot in the image. So the other 17 that we offer are redundant, are robustness to the assay. And this means that you are not depending on each and every part of the transcripts. If some of the RNA is degraded, if some of the RNA is covalently linked to proteins because of formalin fixation, you don't mind, you anyway get the signal. So that makes the assay really robust and makes it good for routine use. What you cannot avoid is that some of these oligos will bind in wrong places. You always will get non-specific hybridization. However, there is no signal to these Z oligos, and a single of these oligos is not able to hold the Christmas tree. It offers not enough base pairs for a strong binding, so the Christmas tree will be washed away and you don't get any noise. So this is the third trick of our technology here. This is how we can really boost signal such tremendously without boosting the background. So in the next slide, you get an idea of the results again. Here we look at the HeLa cell palette that was fixed in formalin and then put on slides. And we stain here with a low copy housekeeping gene, polar2A on the left-hand side. And each of these brown dots, again, represents a single copy of this polar2A transcript. On the right-hand side, we stain with a negative control probe that is DAP-B. It's a bacterial gene, and you see there is no signal whatsoever, so it's really a clean background. Next slide shows you that we not only developed a single Christmas tree that we can build on a target, but we have developed several ones that are independent and that are completely different in sequence. So you can build them in parallel, you can apply them in parallel, and this opens up the door for multiplexing the technology. That is illustrated here in bluish and in greenish. So there's one target loaded with blue Zs and a blue Christmas tree, and the other then in green. And we have 4 different amplifier systems in place. So in theory, you can do up to a 4-plex. In practice, we offer our fluorescent kit as a 3-plex because the 4th channel is in the far infrared and very few people have the right optics on their laser microscopes. So the next slide illustrates this in the 3-plex setting. So 3 different colors. What we use is wavelengths that are similar to FITSI, Psi3 and Psi5. In fact, we don't use these fluorophores because they bleach and are not very stable. We use Atto and what's the other one? Alexa dyes. So anyway, the next slide then shows you what the result looks like in a 4-plex assay. Here we look at the HeLa cell again, and we have a 4-plex staining for 4 different housekeeping genes. The highest expressor in this setting is beta-actin, that's shown in red. And it's so many copies, it's about 1,000 copies per cell, you cannot see individual dots anymore. It's just a red staining all over. The lowest expressor in this list is HPRT1 in green, and you have some 10 to 15 green dots in this HeLa cell. And the other 2 are in between. PPIB is cyclophilin B, that is widely used as a housekeeping gene, and some people also use it as a housekeeper and a control for qPCR. RPLPO is also in yellow, it's a similar expression level. So the next slide now shows you the workflow, and you can recognize that it is extremely similar to IHC, and that was done on purpose. We wanted this assay to fit in a typical pathology workflow. So starting with FFPE material, you do a pre-treatment, you block endogenous, after deparaffinization, you block the endogenous peroxidase activity, you boil the samples to reverse some of the formalin cross-linking, and then you do a protease step to open up the sample and make the RNA accessible for building the Christmas tree. In the middle block, typically you would incubate with antibodies. Here we build our Christmas tree, and on the right-hand side you do the chromogenic reaction, counterstaining, and the whole assay takes about 8 hours in our default protocol. We recommend to do a positive control and a negative control probe next to the target of interest. So the positive control gives signal. If you use a housekeeper, you get signal, and you can be sure that your RNA integrity in the sample is good, and that you don't see a signal for your target probe just because your RNA is too much degraded. Obviously, the negative control gives you you make sure that you have specific signals in the other experiments. So it's also a good measure for pre-treatment. If your pre-treatment is too much, then you get a smeary background, and you know that you have applied, for example, too much protease, or you have boiled your samples too long. So it's also to adjust the pre-treatment conditions. Next slide. So I mentioned already that the assay is highly specific, and this gives you an example here. So here we look at the xenograft sample. So it's a human tumor that was grown on mouse, and in the upper two images we look at the human region from this sample, and the human TGF-beta probe gives a nice signal, whereas the mouse homolog does not. At the bottom two images we look at the mouse region, so the surrounding mouse tissue, and here it's the other way around. No signal from the human probe, but you get a nice signal from the murine TGF-beta probe. So typically the assay can discriminate between rodents and human. If you have 85% homology or less, the assay will discriminate. That is also shown in the next slide, now in a fluorescent readout. Here we look at human papillomavirus, and human papillomavirus is cause of many cancers, head and neck cancer, cervical cancer, for example. And some of the typically used cell lines, like HeLa, for example, have an integration of HPV. Now, there is not a single HPV strain, but there are many different ones, different HPV types. And HeLa, for example, has an integration of HPV-18. If you use the 18 probe to test, you get the red signal here. If you use HPV-16 or HPV-45, you don't get signal from HeLa cells. So here again, we have 85% homology, and the assay can discriminate. What the assay cannot discriminate is between human and monkey, for example. So once you have an homology of 95% or more, the assay will give you the full signal. It cannot discriminate anymore. Next slide. So in this experiment, we have tested if indeed each dot that we see in our image reflects a single copy. And here we have stained HeLa cells for HER2, and we counted 100 HeLa cells, and on average we found 14.4 dots per cell. Then we used the same cell culture, lysed the cells, and measured the HER2 expression level against an in vitro transcript standard curve. And in that setting, we came up with 17 copies per cell on average. So that is really close, and we are confident that indeed a single dot represents a single copy. Next slide. So when you are doing fluorescent readouts and you work on tissues, then you very often are confronted with high amounts of autofluorescence. And I mentioned in the beginning that we have a partnership with PerkinElmer. They developed the nuanced spectral imaging system that can help you to get rid of the autofluorescence. So they have algorithms that calculate the noise and get it out of the image. So that's for tissues especially very, very helpful. Next slide. gives you a nice application. Here we look at a formalin-fixed tissue slide from breast cancer, and it was stained for a cocktail of cytokeratin probes, so we call this a pan-cytokeratin probe, and it stains the breast cancer cells in light blue. And then it was tested for UPA and PI1, that's plasminogen activator. That is one of the diagnostic tests for breast cancer if a patient is plasminogen activator positive or negative. And it was thought that it's the breast cancer cells that have an elevated expression level of plasminogen activator or not. It turns out from this image that it's not the breast cancer cells, but it's stromal cells in the vicinity of the tumor. So it's a very nice example that indeed the tissue context makes all the difference in the world for some biomarkers. And this is also technically, it's a nice example. It was done on FFPE. It used the nuance system and it used the 3-plex assay that we have available. So it's a nice piece of work, and our R&D department is quite proud of this one. Next. So this is the overview and introduction to RNAscope that I wanted to give you so that you have a bit of context where ACD comes from. So Eres called me when they got this results and said that he is about to publish this, and he got some interest from other colleagues, and we started the discussion if it may be a good webinar for the whole zebrafish community. And Eres and his team immediately agreed, yes, we can show our data and explain a bit how we have tweaked the assay to get it to work. And it's my great pleasure that Azadeh and Theresa will now present their their protocol and modifications and their results. And I'm always blown away by these beautiful images when I see them. So Theresa, Azadeh, thanks a lot that you have set this up, that you volunteered to take your time, and take it away.
Okay, thank you, Kai, and thank you all for joining our webinar. My name is Theresa, and what Azadeh and I want to do today is to present our recent publication in BMC Biology, where we describe the establishment of a novel in situ hybridization procedure for RNA detection in zebrafish, a procedure based on the RNAscope technique that was just presented to you by Kai. Our original motivation for establishing this method was the fact that standard in situ techniques for determining the distribution of RNA in zebrafish embryos had several drawbacks. So previous techniques for detecting RNA expression usually rely on the detection of diffusible products of an enzymatic reaction that reduce the spatial resolution of RNA localization and also impair the quantification of RNAs. Usually, double fluorescent in situ hybridization in zebrafish requires 4 to 5 days, since the stainings are performed sequentially and are therefore relatively time-consuming. Last, due to the high temperatures between 60 and 67 degrees used in the conventional assays, colocalization studies between RNAs and proteins cannot be performed as the antigenicity, or in case of fluorescent protein fusions, the fluorescence is damaged by the procedure. So for these reasons, we have established and already applied RNAscope for applications where the conventional assay was not successful. So in the work of Johannes Hartwig in our lab, it was necessary to present the early expression pattern of the RNA encoding for CXCR12A, a chemokine that guides migrating germ cells in the developing embryo to the developing gonad. Using the chromatogenic in situ hybridization technique, CXCR12A RNA expression can first be detected at 6 hours post-fertilization. Using the RNAscope technique, we could detect RNA expression at an earlier time point, an issue that was important for the topics studied in this paper. In the following slides, we will present you with a protocol that we developed for zebrafish embryos using the RNAscope technique and present the advantages it offers compared to standard in situ methods. So again, in this method, special probes are designed as such that they hybridize to the target mRNA, with each probe containing a different tail sequence that provides the base for the assembly of a signal amplifying scaffold. Because this scaffold will only form on a pair of flanking probes, signal specificity is dramatically increased. As a first step, we employed the original RNAscope protocol on zebrafish embryos. And we observed that embryos were damaged in the course of performing the procedure according to the protocol provided by ACD, which was designed for staining thin tissue sections rather than for staining of thick and more fragile whole embryos. Possible causes for the damage can be the conditions such as the high detergent level and salt condition in the buffer provided with the kit. In embryos that survived the procedure, we were able to detect staining of MyoD in the developing muscles, a tissue MyoD RNA is expressed in. However, we had strong background signal in the notochord in both channels of MyoD depicted in red and DAPI shown in green, which is a bacterial mRNA that serves as a negative control. In both channels, we also observed high autofluorescence of the yolk. And last, penetration of the DAPI stain appeared inefficient in labeling all the nuclei, especially in inner parts of the tissue. So given all those issues, we set out to optimize the protocol such that it maintains the structure of the embryos, while solution can still penetrate the tissue and the background signal is kept at a low level. This is the protocol for RNAscope in zebrafish at a glance. You can also find a more detailed version in the paper. In comparison with the standard in situ protocols, fixation times are reduced to 4 hours for very young stages, down to 30 minutes for embryos older than 1 day. Embryos are then dehydrated and stored in methanol. Immediately before starting the procedure, methanol is removed and embryos are simply dried in the air for 30 minutes. Embryos are then digested using pre-treat 3 solution that contains proteinase K before a mix of the probes is added for hybridization overnight. Importantly, points 1 to 4 only require 1 hour of the first day of the procedure. The rest of the protocol is performed by the end of the second day. On the second day, probes can be recovered and embryos are fixed again in a post-fixation step to increase integrity of the embryos. Afterwards, a series of amplification steps build up the amplifying scaffold on top of the probes until at the final step a fluorescent label is added. To counterstain the embryos, we incubated them either in DAPI or in Hüchs solution for an additional night. Otherwise, imaging of the embryos can start directly at the end of the second day procedure. The whole procedure on this day requires usually in total around 7 hours. In our protocol, there are some crucial points we altered when trying to increase the integrity of the embryos and the signal-to-noise ratios. So starting with maintaining the integrity of the embryos, we had to optimize the duration of the fixation time such that the embryos are stable enough to preserve embryo morphology, while to allow effective washing of reagents that if not washed increase the background signal. Embryo integrity was also sensitive to the drying step, as embryos got damaged when either dried for less than 30 minutes or when too much of the liquid was removed, so that embryos in the end stuck together. In the following step, the embryos are digested for no more than 20 minutes, as longer digestion resulted in embryo disintegration in the following steps. Given the short initial fixation time, we included an additional fixation step following the probe hybridization to increase the integrity of the embryo before the embryos are incubated with the provided AMP solutions that present the embryos with harsh buffer conditions. The post-fixation step should not be longer than 10 minutes to allow the efficient washing in the following procedure. The second issue we had to optimize was to increase signal intensity, while at the same time to decrease the background noise. We found that the initial fixation step is not only important for preservation of the embryo integrity, but also for reducing background signal. So the fixation time actually reflects a compromise between increasing the embryo integrity on the one hand and allowing for efficient washing of amplification scaffolds for reducing the background signal on the other hand. To enhance signal intensity in the relatively large whole mount samples, we prolonged probe hybridization from 2 hours in the original protocol to overnight. Also, not only the type of wash buffer, but also increased washing durations dramatically enhanced the signal-to-noise ratio after probe hybridization and the subsequent signal amplification steps. Last, we prolonged the treatment with DAPI to overnight in order to achieve nuclear staining also in the inner parts of the embryo. In the end, the combination of both fixation steps, the use of 0.2x SSCT wash buffer and increased incubation and washing times resulted in optimal preservation of the embryos, while we obtained the best results concerning the signal intensity and background reduction for RNA detection in the whole embryo. So now that we got the protocol for RNAscope to work in zebrafish, we wanted to compare it to other whole mount in situ methods. So we compared the different techniques using probes for MyoD, which is a universal marker for the developing muscles, and is thus expressed in a specific pattern in the trunk and tail region of zebrafish embryos. So RNA detection using the chromogenic in situ method has the disadvantage that the colored precipitate interferes with imaging of deeper parts of the tissue. Employing the fluorescence in situ technique, MyoD expression can be visualized at a better resolution. In addition to the standard fluorescence in situ protocol, we also examined an optimized assay using self-made TSA substrates that resulted in brighter signal for MyoD RNA and reduced background in the yolk compared to the standard protocol, while the spatial resolution appears relatively lower. When using RNAscope, we obtained a staining that appeared to be more distinct and to reflect more accurately the subcellular localization of MyoD RNA in comparison to the standard and optimized protocol, which appear more aggregated and diffuse. So after obtaining these results that suggest that RNAscope offers high resolution in RNA detection, we wanted to evaluate this technique in detecting subcellular localization patterns. We probed for VASA RNA in 4-cell stage embryos, where it is localized to the cleavage planes of cell division, and also high-resolution imaging of the myotomes shows distinct localization of MyoD RNA, labeled in red, within the cytoplasm around the blue labeled nuclei of the developing muscle cells. So in summary, RNA detection using RNAscope is a good alternative to the existing fish techniques, but it has clear advantages, such as a fast and easy protocol together with low background and the capacity to detect subcellularly localized RNAs. The next point we wanted to check is if RNAscope would allow to detect multiple transcripts at the same time in the whole mount zebrafish embryos. So here you can see a 12-hour-old embryo in which the RNAscope protocol allows visualizing the RNA expression patterns of CXCL12A and lateral mesodermal tissues in green, together with VASA RNA expressed in primordial germ cells in magenta, as well as the RNA of MyoD that is expressed in the developing somatic mesoderm labeled in red. In this example, we have therefore successfully visualized 3 RNAs at the same time. Importantly, RNAscope-based detection uses a signal amplification scaffold instead of enzymatic reactions, which produce diffusible products. This feature of the protocol allows reliable quantification of the signals emitted by the tissue and thus of the amount of RNA expressed in this tissue. So in this example, we measured signal intensities of CXCL12A expression pattern along the white line, revealing that PGCs localized to positions in the embryo where the RNA encoding for the chemokine CXCL12 is expressed at higher levels. This feature of RNAscope to allow the simultaneous detection of multiple RNAs at a resolution which facilitates quantification of the signals in whole mount embryos, we think is an important and useful tool for developmental and cell biologists. So next, we tested the sensitivity of RNAscope to detect transcripts expressed at low levels within the embryo, which is now what Azadeh is going to talk about.
Hi, thank you, Teresa. I'm Azadeh, and I will start with telling you about how RNAscope protocol can be used to detect rare transcripts. So we examined the RNA expression pattern of CXCR4B, the receptor for the chemokine CXCL12. This is expressed in PGCs, in the lateral line, the nose, and in cells in the hindbrain of 24-hour embryos. We consider the expression of CXCR4B as weakly expressed in the PGCs, since using short staining reaction in a chromogenic in situ procedure, we failed to detect transcripts in those cells. Only after a long staining reaction, CXCR4B RNA becomes visible within the region of the developing gonad. So using RNAscope, we were able to clearly detect CXCR4B RNA in PGCs, which we confirmed with co-labeling the cells with the PGC marker nanos. And thus, the RNAscope not only allows specific labeling of multiple RNAs and the possibility to detect subcellular RNA localization patterns, but also offers a high sensitivity that allows quantification of signals and detection of rarely expressed transcripts. Another important feature missing in current in situ hybridization techniques is the ability to detect proteins in the tissue. So transgenic fish lines are powerful tools in zebrafish to address many biological questions. And combining the visualization of protein detection with standard in situ hybridization technique is problematic, since standard RNA detection methods require incubations at temperatures as high as 60 to 67 degrees. Such high temperatures destroy the fluorescent proteins, and in many cases the antigenicity as well. The relatively low temperatures used during the RNAscope protocol, up to 40 degrees, could in principle preserve the function of proteins. To test this idea, we used 24-hour zebrafish transgenic embryos whose PGCs express EGFP on their plasma membrane. Indeed, VASA transcripts could be detected in the cytoplasm of the PGCs together with a strong EGFP signal on their membrane, which was preserved through the RNAscope procedure. Similarly, we used transgenic embryos in which DS red was expressed within characteristic primordial germ cell perinuclear structures. DS red expressed in these PGCs, in these PGC granules, could be detected along the VASA RNA, clearly presenting VASA RNA localization relative to the granules within PGCs. Increasing the hybridization temperature to 50 degrees, which is compatible with the RNA detection, negatively affected protein fluorescence. Therefore, for protein detection during the RNAscope procedure, maintaining the temperature of 40 degrees is required. Along the same line, we could visualize MyoD RNA together with a cadherin labeled by antibodies directed against the protein. RNAscope technology is thus compatible with immunohistochemistry experiments in whole mount embryos, where protein localization is determined by use of specific antibodies. Investigating the RNA expression pattern in internal organs of the embryos of more advanced developmental stages is often required. Therefore, we evaluated the RNAscope method in this context. For that, we use one-day-old transgenic embryos that express EGFP protein in neuronal cells under the ELAP promoter. And the EGFP fluorescence was preserved during the RNA detection procedure, in which the RNA encoding for the protein, which is the RNA for EGFP, could be localized as well at different tissue depths. To demonstrate the same point, on older embryos, we examined the RNAscope technique on 3-day-old embryos, in which cells in the vasculature system were expressing EGFP RNA under the control of the flea promoter. The endogenous EGFP fluorescence and EGFP RNA were detected comparably at different tissue depths, as you can see in internal vessels labeled by the white arrows. Z projections of all optical sections you can see on the right, and the graph shows the signal ratio of EGFP RNA to EGFP protein fluorescence measured in central parts of the embryo at different depths of the tissue, indicating uniform labeling independent of section depth. So this feature allows the 3D visualization of the embryo, showing the expression pattern of both the protein and the RNA. Together, we could show that the RNAscope method allows the detection of RNA molecules in a wide range of developmental stages and tissue depths in whole mount zebrafish embryos, consistent with the idea that all the reagents used can penetrate the tissue. An important feature of the RNAscope method is that it provides very high signal-to-noise ratios. As you can see here, by dividing the average pixel intensity of an area which expresses EGFB with that in a region where EGFB is not expressed, we could show that the optimized RNAscope protocol provide an excellent signal-to-noise ratio. When optimizing the protocol for new probes or for use in different organisms, the following considerations can be taken. So during the optimization procedure, we found that the hybridization temperature had a critical effect on the signal-to-noise ratio. For example, a complete lack of signal was observed when employing the standard 65 degrees hybridization temperature used in standard fluorescent in situ hybridization methods in zebrafish. Using a 55 degrees for the hybridization resulted in low signal-to-noise ratio, while using 60 degrees resulted in low signal as compared with the standard 50 degrees and 40 degrees temperature that we suggest to use in the RNAscope protocol. Also, increasing the hybridization temperature to 50 degrees negatively affected the survival of the protein fluorescence. Another parameter to consider when optimizing the method is the duration of the hybridization. To further enhance the signal in the thick embryo samples, we prolonged the duration of the hybridization from 2 hours in the original protocol to overnight, which indeed yielded an enhanced specific signal. Similar modifications could also improve the results for specific probes at specific developmental stages and in embryos of different species. So another critical step was the duration of fixation, which was important not only for preservation of embryo integrity, but also for reducing the background signal. We found that reducing the fixation time to 30 minutes at room temperature in case of 24-hour embryos significantly enhanced signal specificity as compared with the 24-hour fixation at 4 degrees. So in summary, we have presented an optimized protocol for the use of RNAscope in zebrafish whole mount embryos for detecting multiple transcripts at different tissue depths and in different developmental stages. We could also show that the protocol is compatible with the determination of protein localization in embryos. The improved protocol preserves embryo integrity and is remarkably rapid and can be performed in less than 2 days for multi-transcript labeling. The uniquely designed probes for RNAscope and the signal amplification system allow the detection of RNAs which are expressed at low levels. Also, the exclusion of antibodies contributed to increased resolution and excellent signal-to-noise ratios. These features allow quantitative evaluation of transcript levels in the embryo. Finally, the fact that the label is confined to the position where the RNA resides allows high-resolution detection of the transcripts, facilitating the determination of subcellular localization of the molecules. We believe that minor modifications to this protocol would allow its application in embryos of other species and also large tissue samples. For questions, you can contact this email address, and we will be happy to answer your questions.
Yes, fantastic. Thank you. The assay performance in your specific samples. We And with this, I would maybe also start from the RNAscope part on the next RNAscope concept of. You need probes, and we can design any gene for zebrafish and other species. Meanwhile, we have a list of 4,500 inventory probes, but we could create a new one within 2 or 3 weeks. We also provide control probes, and you have the DAP-B negative probe that is specific, and we will deliver 3 different positive controls for zebrafish, and that is the MyoD, the GSC, and OC1, if I remember correctly. So this is also available from the tech note that we will put together. I mentioned that we have different versions of the assay of the reagents. We have chromogenic readouts that were primarily for formalin-fixed stuff. What the group of Iris Rasius, so other and there is the multiplex fluorescent assay, which comes as a 3-plex. For our slide-based incubations, we recommend to use this hybridization oven that you can see in the image here. I know that Azadeh and Teresa used a different way to incubate. Maybe then
we can hear you.
We can hear you.
Okay, so maybe to the question of the incubation. So we performed the staining in standard Eppendorf tubes and incubated them in normal water bath. We didn't use the hybridization oven. Yeah.
So the experiments where we did the immunostainings, basically we did the RNAscope procedure on the embryos first, and then this was followed by physical sectioning of the embryos and then antibody staining on them. So there we did not have, we only had in the permeabilization solutions, which included tritone. But we did not have any proteinase K treatments within this procedure.
in the immunostaining procedure or in the RNAscope procedure?
In the RNAscope procedure, the digestion goes through pretreat 3 or pretreat 4, which works as nice as basically the same. But for the immunostainings, there was not a proteinase K treatment involved.
Okay, yeah, but these pre-treatment reagents include protease. So you did some. Yes, so we have other. Yeah, so we have other customers that also tried to combine immunohistochemistry with RNAscope, and we have many reported successful experiments where people did RNAscope followed by IHC, and as our pre-treatment includes a protease step, sometimes the epitope is damaged and it doesn't work, but very often it does work very nicely. Maybe this is also an answer to this question. Then here the next question is, a new method for ISH DNA HCR was reported recently with hybridization temperatures of 45 degrees. What are the advantages of the RNAscope over that method? So
I think we also cited this new method in our paper. We haven't tried this method as an alternative option, so we can't say if it's better or worse. But what we just realized is that it has a relatively long protocol, the HCR technique.
okay. Here's another question. In your experiment on zebrafish, did you use only 2 probes for a single gene or more probes, as suggested at the start by Kai? Is the use of more than 2 probes per gene only required for specificity control? No, so for the zebrafish probes, also ACD did the design, so we used our default design, and that is 20 pairs. So the presentation of Azadeh and Teresa simplified this a bit. They only showed a single of these branched scaffolds, but in fact it is more than just a single one. It's more than just 1 pair of oligos. It's more, and it depends a little bit on the size of the transcript. If you have a transcript that is long, then we do all the full 20. Sometimes we deal with shorter transcripts that maybe only offer 400 bases, and then we have to live with a little bit less, and that is about the threshold. If the transcript is shorter than 400, then it gets tricky to get enough sensitivity for single copy detection. If you are above 400, then usually it's no problem, and we can guarantee the probe to work. It also depends a bit on the sample quality. So when you deal with clinical samples where you very often have RNA degradation, then it's safer to have a longer transcript targeted. If you have good quality samples from translational medicine or potentially from zebrafish, where you are in full charge of the sample preparation, the embryo preparation, then probably RNA quality is really good, and then you get away with shorter transcripts and still get the signal, as you were shown here from these great results. Next question here: would it be possible to design probe-specific for different splice forms of a given transcript? That relates to my former answer. It depends on the difference between the 2 splice variants. So if the extra exon is only 30 bases, then we cannot do it. If the difference is like 400 bases, yes, then we can do it. So it's for sure it's worth having our bioinformatics experts have a look at the sequences, and then they come up with a suggestion if it's possible, for sure, if it's not possible at all, or if it's a maybe. So we can discuss these kind of questions on an individual basis. You mentioned about non-coding RNA detection. How does it work on microRNA, which is short, 22 nucleotides? There may not be many probes that you can bind to it. Yeah, that's true. So something like microRNA, we cannot address with the current version of our assay. They are indeed too short. As I said, the threshold is something like 400 bases. If it's what we have done successfully, we have looked successfully at precursors from microRNA, so stuff that is like 500, 600, 800 nucleotides long. MicroRNA at this stage is not in our portfolio, so to speak. What else do we have here? For zebrafish, have you tried in later stage fish, like 6 or 7 days post-fertility? Have you tried older stages of embryos, Azadeh? Teresa?
So, the latest time point we have checked has been 4 days post-fertilization. Older stages than this we haven't tried.
Okay. The next question here is the protocol compatible with bleaching the embryos for eliminating the pigmentation?
So the pigmentation of the zebrafish embryos for older stages, we do through PTU, incubation of the embryos in PTU. This is basically, this will be done before fixation of the embryos. So if this is what they meant, it doesn't interfere with the procedure of the RNAscope at all.
Okay, so you do this depigmentation first, and then you follow with the RNAscope protocol, so no problems. I see. Any idea why the longer fixed embryos showed such high backgrounds? So from our own experiments, we know that if you have too much fixation, then these scaffolds get captured in this network of crosslinks, and then it's difficult to wash them out effectively, especially when it's very thick sections. So that's probably the cause that then they are kind of trapped in the tissue, and then they also load the labels, and then it's unspecific. You don't get specific dots, but the overall signal goes up, and that seems to be the problem. But maybe you can comment from your experience as well.
I think what you said is actually true. So what we experienced was the same, that basically with longer fixation times we get increased background, and we could get rid of partly with increasing the washing steps and the duration of the washing, and I think that was probably the reason.
Yeah. Yeah, so you have to imagine that these pre-amplifier and amplifier DNA molecules are quite long. It's not an ordinary oligo like a 50-mer. They are several hundreds of nucleotides long, so they can easily be trapped if they have too much they can stick to. That if you have a huge, a thick sample like a whole mount, then that's creating more problems than if you have a thin one. Another question here is, have you tried it on whole mount isolated organs from adult or juvenile fish?
So no, we haven't tried it. So we also usually doesn't work with extracted organs from older stages, but yeah, maybe it needs just some modification of the protocol we provide to get this also running in such tissues.
Yes, right. So we have customers that tried whole mount tissue sections, like 100 micron sections from mouse or from other species or mammalian species, and they successfully got good results from that. So maybe that's the closest answer to your question here. It should work. Maybe it requires a bit of tweaking of the protocol in a different way. So they are probably not as fragile as the embryos. So probably you can stay closer to the standard RNAscope manual than for the zebrafish embryos here. So I have one last question here that I find. How long can you store your fixed embryos in methanol?
So the procedure is actually like the other in situ. So at least one overnight it goes through methanol. It will be basically kept stored at -20 degrees for one overnight.
And how long can you stay them there so they are still stable and usable? Can you store them for a longer time?
They are in principle stable and usable, but we had this experience that as the embryos they get stored longer, the background detected in the samples gets higher. So we did not. At some point we stopped using embryos which were stored more than 2 weeks.
Okay, so 2 weeks is kind of a threshold for you. Great. Thanks a lot. I want to close the question and answer. Our time is over. There were 2 or 3 questions that asked for price information. So I would ask you, please go to this website, fill out the form, get in touch with us, and you get an individual pricing. So when we started, or not when we started, when Ares had submitted his paper, he also shared some data with colleagues, and he said some are really excited, but they said the cost of this assay is really high, and yeah, it's not an inexpensive technology, I have to admit. Ares said he wanted to give his colleagues in the Zebrafish network access to the technology, and Zebrafish probably have less budgets, so Zebrafish researchers have less budgets than the clinical research, and I raised this idea with our management team, and we finally came to the decision, yes, we want to give this technology to the zebrafish community, and also as an appreciation of this great piece of work that the group has done. And so we will make a promotional pricing, which is half the price, available to the zebrafish community. So the conditions are that you have to include a zebrafish probe, target or control probe, in your purchase. And for us, it doesn't make a difference if you go with a fluorescent kit, just like Azadeh and Teresa, or if you prefer a chromogenic readout, that doesn't matter. You can have any of these variations. But it must include a zebrafish probe, and we will run this promotion until almost until end of the year as a start, and then we will have a look if it was worthwhile. So Eris promised me there will be so many people excited about this and they use it, so you get lower price, lower margin, but you get the volume, and hopefully that compensates. So we will have to do the math at the end of the year then. In any case, what I really appreciate is then that Teresa and Azadeh volunteered to answer further questions. So if any other question comes to mind, please send an email to RNAscope for zebrafish at icdbio.com. That will be forwarded to Azadeh and Teresa. But please consult the tech note and our manual first. You can imagine that these two ladies have more important things to do than support the global zebrafish community. So I really appreciate that they volunteered here, but it will take a couple of days until you get an answer, so depending on their priorities. And yeah, please have a look at the tech note first and at their paper. Probably a lot of questions can be answered by that means. And yeah, and to get this kind of pricing, fill in the form that we have your contact details, and we will contact you and give you an individual quote for what exactly you need. So thanks a lot. Thanks everybody. Go to our website. You'll learn more about RNAscope. Have a look at this fantastic paper. And on the website, you will also see that we have distributors in many countries. So if you are not in the United States or in countries where we work directly, we have distributors